Today we will discuss about a new paper where the idea of that paper is that if they can modify these antibodies which are pathogenic in nature and cause autoimmune inflammations in a way that by adding this sialic acid and galactose, the nature of this pathogenic antibody will changes into the anti-inflammatory antibodies and will suppress the autoimmune response. So how these things has been achieved? These things has been achieved by glyco engineering, glyco engineered glyco by engineered glycotransferase in a way that it will add sialic acids and galactose, which are less inflammatory uh, in nature, to the pathogenic antibodies, and thereby it will uh, thereby the final outcome will have the antibodies that is less inflammatory in nature. So in order to work on this hypothesis, what they did is they fused they fused this glycosyltransferase into the Fc portion of this human IgG, and that is shown in this panel C, where what they have did is they fuse this B4 GalT1, and which they termed it as B4FC, and when they and another glycosyltransferase, that is ST6-GAL1, they fused ST6-GAL1 and termed it as ST6FC. And in the panel B we can see that the structures in the let's start with panel A. In the panel A is the human IgG, where at N297 there is a N-glycosylation site, that is NXT site, and in this NXT site the representation of the glycans is shown in the panel B, where the box portion shown is shown as the anti-inflammatory glycan structures, and its inflammation can be reduced by adding this fucose or this galactose, and this by adding this fucose and N-acetylglucosamine at this site, and also by adding of this galactose and sialic acid, the anti-inflammatory response will be reduced. And so the idea to introduce this kind of glycan structures is through the addition of the glycosyltransferase to the human IgG, so that the reactions can occur, and it will cause the addition of the additional required glycan structures at this glycan site. So first, they in order to examine in order to examine their engineered human IgGs, they did the in vitro experiment with the fatuine. So first they treat the fatuine so that they will have a glycosylated fatuine and also the glycosylated fatuine, and then use this aglycosylated fatuine with this B4FC and this acceptors, that is UTP galactose, to see whether the galactose is again installed in the again again present or again being added to this G0 fatuine, which does not have a glycosylated. And here we can see that in the G0 we do not see the signal for a glycosylation or terminal beta 4 galactose, whereas when we add this B4FC and UDP galactose, we can see that the signals for UDP, we can see the signal for terminal beta 1,4 galactose. And for the G2, which has this galactosylated glycan structures, and the representation is shown on the bottom right corner, what is G2 and what is G0. So in the G2, which has the galactos, galactosylated fatuine, when it is treated with this ST6FC and CMP sialic acid, the galactosylated will be uh, the sialic acid will be added to the alpha 26 position. So that can be seen in the middle row, where terminal alpha 2 sialic acids can be seen using the SNA lectin blotting. And thereafter when they add both the B4FC and ST6FC, what they have seen is that there is a presence of this beta 1,4 galactose in the G0 fatuine, and when they also use the CMP sialic acid, they can see that the CMP sialic acid is also added to this galactose, which is reflected by the lectin blotting of alpha 2,6 sialic acid, as the sialic acid is added after galactose. So in the final stage, it will be reflected by alpha 2,6 sialic acids or SNA lectin. And when they did this experiment also on the mouse model, they have find out is that the mouse models and the human mouse FC and human FC also shows the similar results, in a way that in the mouse FC with the mouse FC with this engineered uh, antibody, they can see the formation of this terminal beta 1,4 galactose, which is checked by ECL lectins when they added B4FC and ST6FC along with this UDP galactose. However, when they added CMP sialic acid also, they see that sialic acid is also attached to the galactose, that is shown by this terminal alpha 2 sialic acid. And some of the incomplete reactions, because of the incomplete reactions, they can also see terminal beta 1,4 galactose. And similar results they have observed for the human FC, engineered human FC also. So this indicates that the engineered IgG can cause or can help in suppression of the anti-inflammations by addition of the sialic acids and galactose, which are anti-inflammatory, in nature, and can help in suppression of the autoimmune response. So that's all for this video. I hope you understood their engineer design and how they validated their design, and what is the hypothesis of the paper on which it worked upon. If you need to understand more about their engineered human IgG or their hypothesis of the paper, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.